it's my pleasure to introduce Stefano Allesina, who is going to give us a next talk, and he's going to talk to us about computing skills for biologists on building a toolbox. And Stefano is on the faculty at the University of Chicago. He is really an expert on network science and methods, particularly in trophic programs, and he's also one of the holders of one of the Burroughs Wellcome Fund projects. So, Stefano, it's all yours.
Thank you. Thank you for like your presentation. I thought of talking about computing because I thought it would be complementing what you were discussing about mathematics. And so just like to say a few words about me, you already said some. Theoretical ecologist at the University of Chicago. Strangely enough, I studied actually environmental sciences in Italy at the University of Parma, where in 1999, I learned how to program in Pascal of all languages, like when dinosaurs roamed the earth. And this opened like the world of science for me. And in fact, I became a scientist basically because of this one class that I enjoyed so much learning how to program. And then in fact, I went on to work as a programmer for a small telephone company while I was waiting to start my PhD. And I learned a lot. And right now, like my lab mostly uses R, Python, and C, but I'm well versed in a bunch of other programming languages. So I just wanted to share some of my adventures in graduate education, and especially when it comes to computing. And I would like to talk first about like a self-selected group of students, or really a class. And then later on, how to teach computing generally to all incoming PhD students, which is, as you can imagine, a very different problem. And this will bring us actually to the wonderful time before COVID, where people could like work together in the same room. And so when we're thinking about like computing, we're actually thinking about a bunch of different aspects, right? And one that I would like to stress, which was actually apparent like in Lou's presentation, is like the heterogeneity of the incoming graduate students like across like biology, and the fact that this is a constantly changing landscape, right? What is deemed important or not, as we were discussing before, changes, and like the techniques change. So we have to constantly adapt to this thing, especially to do what? To ensure that our analysis is correct, that is reproducible, to possibly expand our scientific horizon by learning new things. We have new ideas. And in this way, we can, for example, accelerate discovery. So we have all of these different ideas in mind, but what can we do to really like get people started in this area of computing for biology? And so here I would like to detail my thinking behind the class that is called Computing Skills for Biologists. And the idea is to target PhD students in biology across the board. So I'm talking people doing cancer biology and paleontologists in the same room, learning together about how to compute and how to use these machines to do science, right? And of course, you can only cover so much, especially at the University of Chicago, where we're on a quarter system, which means 10 lectures, 3 hours each, so 30 hours in total. And the way I'm trying to do this is like to have about 20 students in a computer lab working on their computer while we are having class, right? And my goal really is just to give people a sense of these building blocks that can be used to create like pipelines that can automate the data collection, the data analysis, like the presentation of figures and whatnot. And so like my goal is that at the end of the class, the students will have a toolbox, which differently from what you get if you buy a toolbox on Amazon, includes all the tools, right? So they've seen these tools and they know how to use them. Of course, what we could do is like to be very academic and go very deeply in each and every tool, but then we could cover only a few realistically, or we could be much more like shallow and present a variety of tools. And the way I chose to do is exactly this way, in which I present a variety of tools. I use real data from published paper for all the examples and exercises. And then because we have to go quickly through these tools, basically I want to give you a sense of why would you want to learn this tool. I want to give you a short introduction to what is this tool about, have some exercises, and these exercises ramp up in difficulty quite quickly. And then finally, you have a reading list where you can really like go deeper in the tool if you think this would be useful for your research. And of course, like you have to choose a variety of tools. I chose, like, according to my own priorities. I didn't do a survey as Lou was doing before to decide what to teach, but these cover like very basic things. For example, like the Unix shell, right, that you can use to automate like data pipelines, version control, which I think should be taught to every graduate student because it can save the day in many, many occasions. And then we start programming. And for programming, I chose to teach Python because I think it's a very well thought out like language, and it's easier to teach how to program well in Python. How to write good code, meaning that is correct, first of all, and I introduced debugging and unit testing, but also that other people can read and understand, because we're scientists, and science is like a multinational endeavor, and you have to be able to share your work with other people. Regular expressions for text, and then scientific computing. What are the tools that are available for doing things that we do as scientists every day? For example, integrate differential equation, compute difficult integrals, work with large data sets, work with sequence data, these type of things. Typesetting, how to write your thesis in LaTeX. Computing for statistics, I introduce R. So just to give like people the sense that once you learn one language, it's quite easy actually to pick up another language. In R, I stress very much like data wrangling and visualization, right? How to massage your data such that it's ready for analysis. How do you organize this using tidyverse? And finally, I have some notion of relational databases. So these are 10 tools. You could choose 10 different tools, and I think it would be fine anyway. And it is a bit of a challenge to present so much material to the students in such a short time. And so what we decided to do is more of a flipped classroom type of thing. So I have very, very extensive notes that the student read at their own pace before class. And then each class starts with a brief recap, right? Like say half an hour where I take the summary of what we should have learned if you read the chapter carefully, and I give some basic example. And then I lead the class to the practical solution online. Like when we're doing this thing, like we are solving the problem live, right? We're live coding and we solve problems of increasing like complexity. And then at the end, I assign homework, which is actually quite difficult because all the homework is, for example, take this paper, replicate figure 3. So you have to read the paper, understand what they're trying to do, get the data that is provided, massage the data, plot the results, or do the statistics, or do what you have to do. The homework is not great because what I'm trying to do is to really like foster like this kind of self-discipline of trying to learn for the sake of learning because it's useful for you as a scientist. And the only thing that is graded is a final project, which I'm going to explain in a little bit. But the idea throughout the class is to show that these tools are essential, really, to solve important problems in biology and to boost the confidence of the students, because like by the time the class has ended, they have solved quite complicated problems and they've created quite complicated pieces of software, and they can find like some sort of pride in seeing how much did they progress in these 30 hours of class. Again, I encourage discussion and collaboration. I always ask students for possible solutions. We discuss the pros and cons of every solution. And I try to make these tools immediately applicable by showing that these tools are actually used to solve problems that you need for papers, for your science. Just to give you an example of some sort of like warm-up exercises, this would be at the end of the recap. For example, take all the data on Nobel laureates, and now find what is the most common surname of the winners of the Nobel Prize, or find which Nobel Prizes have been awarded, because some years we didn't award all the Nobel Prizes. And so if you do this type of thing, you should find that certain scientists, for example, Marie Curie and Linus Pauling, got like multiple Nobel Prizes. Of course, not always for the same discipline. Another type of exercise to work with large data in R, in dplyr. Download like a file from the Social Security Administration with all the baby names given to Americans from 1880 to 2017. Now you can massage this data into like something that you can work with. And now what I try to ask students is to find names that have changed dramatically in frequencies due to some specific event. For example, if you see here Elvis, and there's two peaks in the frequency with which babies are named Elvis, and one of them is the debut of Elvis Presley, and one is the death. And then if you look at Osama, of course, like this frequency decreased dramatically after 9/11. And then if you see like Hermione, this was basically not a name until like the first Harry Potter movie. Or if you look at Khaleesi, which is an invented name, only pick up like in frequency after Game of Thrones, right? So maybe they should have seen the whole series before deciding. And so what are the outcomes of this type of activity? Well, first of all, I want to stress like this idea of final project to make things directly applicable to the students. What I asked them to do is to pick up a problem in their own laboratory, something that people spend time doing and it's boring or it's difficult or requires a lot of work, and completely automate this process such that they can save time for them and for their lab mates. And what is one of the perks of the class is to see how many different problems people have and how inventive are they like to solve this type of problems. And I'm actually very happy to report that some of these problems actually made it into publication. So this data co-regulation data harvester for Tetrahymena thermophila was one of the final projects of actually an undergraduate student, Lev Tsipin, that now is a graduate student at Caltech, that started during this class. And then, of course, he worked on it for another 1,000 hours. But like the comments here on the left show you that really like what I'm trying to do is to really boost confidence and empower people to try to learn new computational skill. And once you know how to learn a few things, it's much easier to learn the next set, right? So that's my goal. At some point, Madeleine Wilms, who was like a postdoc in Joy Bergelson lab here at the university, took the class as a postdoc, and she enjoyed it a lot, so much so that then she wrote me saying, we should turn this thing into a book, right? And at that time, I had basically 400 pages of notes, for well-organized notes, with code, et cetera, how hard can it be? So this was in 2014, and 1,615 days later, you can buy like the book on Amazon. So, and it's actually 400 pages. So we basically rewrote the same 400 pages several times, just to say that if you're thinking of writing a book, you should think again, because it's a lot more work than you would give it credit for. And again, the approach that we're trying to follow is the same of the class. Each chapter starts with like something, what is Unix? What is version control? Why would you want to learn this? And I'm trying to mimicking this idea of like these warm-up exercises that we have in class as as these sort of intermezzos, like these short exercises that you can do while reading the material. And then exercises at the end of the chapter range from fairly simple, like the first two, and then they ramp up in complexity. And again, they only use real data from real papers. I stress the use of free software. So all the software that we're using is freely available to everybody, because then people from all over the world can contribute. And then I provide, of course, full solutions online, both in terms of like pseudocode, the actual code, the discussion of why did I choose this strategy and whatnot. And there is a website where there's even more material. Okay, so but these are students that decide themselves that they want to learn something about computing for biology. What if I want to reach a much wider audience? For example, all of the incoming biology PhD students at the University of Chicago, which are about 100 per year. So this is work done in collaboration with Stephanie Palmer, who is a neuroscientist here, and Victoria Prince, who is like a developmental biologist. And our main issue is that we're facing, we think, is like try to ensure like that our students are able to write reproducible research, right? Reproducible analysis of data. And this, we know, it's a huge problem in the sciences, and it's become like, it came to the fore fairly recently as one of the main problems, in fact, that we have in science. And if you read like the literature, there are very many articles trying to advocate for best practices for scientific computing. This is coupled with the problem of reproducibility, and this is a very interesting article from Nature where they were asking people whether we are in a reproducibility crisis and whether they have attempted and failed or succeeded in replicating other people's experiments. It turns out that the picture that is painted is not like the greatest. We would like to do much better than this. And so what we want to do is to combine basically three main aspects. These are all incoming students in biology, so they haven't even started like their PhD yet. It's like the first week that they're on campus. And we want to introduce them to computing mostly we cannot cover that much material, right, in 1 week before like they start graduate school, but it's more of a springboard, like give them like some sort of pointers to start learning, about computing, stressing what best practices for writing, testing, and publishing code, and then show that these skills are really necessary to do any good work in biology, that this quantitative analysis and modeling is so ubiquitous in biology that you cannot escape it, and if you cannot get rid of it, you should at least learn about it. And then couple this with the introductions to techniques that to ensure that analysis is correct, discuss the main features leading to what is a reproducibility crisis, and the use of big data with like the perils and joys of big data. And then because we have like this captive population, we want also to start our way into professional development, how to start graduate school with the right attitude, make friends across like different disciplines, learn about resources and research opportunities at the university. What we end up choosing as a strategy is a fairly involved and fairly expensive, actually, strategy, which is a residential bootcamp. So we will have an intensive, fast-paced introduction to quantitative methods in biology with, as I said, an emphasis on computing, again, using real data. Often the data are generated by the instructors themselves, so they can really speak passionately about these problems, and then show that the same techniques find applications across like all of biology. We used R, Python was the other finalist, and then lectures, we start with some sort of tutorials on how to program and things like that, and then they ramp up quite a bit in difficulty. All the materials are available. In fact, I want to share them with you, and I want to discuss this type of thing at the breakout sessions. For the location, this is pre-COVID. We actually went to the Marine Biological Laboratory, which is affiliated with the University of Chicago in Woods Hole, Massachusetts. And this is like a marvelous place. And so here we can think of how good it was when we could travel. And this is like a fantastic place also for research. There were 58 Nobel Prize winners that worked at NBL, and it has a fantastic history. So, take all incoming PhD students. You can see here that it was before socially distancing with each other. Bring them to the same place, divide them into groups, because there's no way you can teach like 90 people at the same time how to program. So they rotate through these different activities. These students come from very broad disciplinary interest and very broad range of backgrounds, from people that never use the computer besides like maybe checking Twitter, to people that have a degree in computer science. And as I said, what we want to do is to split them into groups. We give each group a mascot, which is a model organism going from Arabidopsis here to Drosophila to other more strange like mascots, for example, Tiktaalik rose, which is like one of the first transition between like aquatic and land animals, which was discovered by one of our faculty. And so students are assigned to these groups, which are 12 like the tribes of Israel. They work with their group to solve like challenges that we give them daily, and like whoever does the best is celebrated at the end of the bootcamp as the best like team. We have team building activities on arrival. We have some sort of reflection on group dynamics at the end. The students produce their own little video to say how did it go to work with other people. To foster this kind of like esprit du corps, we provide people with like temporary tattoos that they can exchange with their own organisms. And the students have a lot of fun with these. All right, so just like in terms of organization, a full day for each day goes from 8:30 a.m. to 9 p.m. We have like some morning or evening off for they rotate among groups. It's a full week, so we start on Sunday, we depart on Sunday, and each activity includes between 2 and 4 groups, hence the choice of 12 groups to begin with. We have actually such a complicated schedule, because we have to move these 12 groups between different rooms, that what they ended up doing is a software that builds the schedule. And in fact, I can prove that it maximizes the entropy, like the number of times people meet each other is maximized. And so we do some sort of introduction to R. We do some imaging, because the NBL is fantastic, like microscopy facilities, and is very famous for that. And then we talk about defensive programming, visualization, reproducibility of data analysis, and then in the end, some statistics. So this is how it looks, right? These are like two groups working together. And so you can see it's a mix of like board, programming live, and working with their groups. And finally, we have some workshops, which instead of being like generic for all the things, they're specific to a discipline. So we have had the workshop on neuroscience, infectious disease modeling, microbiome dynamics, population genetics, next generation sequencing and whatnot. And so here we bring some faculty who has a very specific expertise, and their goal is to showcase that even in their discipline, you cannot really get by without some serious computing, right? So to stress again that whatever you do in science, you're going to do it in front of a computer. So you better deal with this. So here's, for example, one of the workshops led by John Novembre on population genetics. Because we're at NBL, we also have some activities that are connected with the Marine Biological Lab. For example, we host like talks from NBL faculty to showcase the opportunities for research there. We tour the Resource Center, and we even bring the students on a collection boat so we can take pictures like this with people with horseshoe crabs or like looking at other aquatic organisms and growing an appreciation for biology. When we go back on campus, we think that we need to hammer them again with this type of material. So we revamped the mandatory ethics course into a much longer course. And this was extended to introduce modules on data analysis and reproducibility. In practice, what we will have always is an R refresher that is focused on writing good code that other scientists can read, a refresher on unit testing, how to make sure that your code is correct, and then on automation of code publishing, right? How to make sure that you can share this code very easily with your colleagues. And finally, on data storage, organization, and publishing, even with the idea of preservation in the long term. So we even involve like the library because they have like expertise on preservation of data for decades. And just to give you like a sense, this is we probe the students with quizzes before and after. So these are paired samples, so we can see whether people have learned something by taking this course. So we probe them on basic computing, basic statistics, ideas about reproducible research and best practices. And we repeat this same type of quiz, even though with different questions, after a year and then after 2 years. So we can see how much of this is retained. And we're lucky that because we started this, we could probe with the same questions students that did not take the boot camp. So we have some sort of natural control group. And then, of course, we have evaluations from the students. We even have a set of like people that are observers that come and record our lectures and then give us some sort of input on like teaching methods and things like that. Of course, like this cost quite a lot of money, actually. So it was initially funded as one of these initiatives to bring together NBL and UChicago, then was partially funded by the National Science Foundation with an Innovation in Graduate Education grant to Victoria Prince, me, and Stephanie. And then each graduate program actually contributes a certain amount per student. And we're still like thinking about long-term solution, which could involve like some donors or some other more like a long-term source of funding. All right, so that's what I wanted just to give you a sense of like, what are the challenges that we're facing when we're teaching a selected group of students that have a very heterogeneous background? What are the challenges that we face when we're trying to broaden these and teach to all students in biology? And I have a bunch of resources here that I will bring up also in the breakout room so we can discuss this. And I have similar type of material for when it comes to statistics and more mathematically oriented education, but I just wanted to focus on computing here. Thank you.
Thank you, Stefano. There has been a active set of comments in the chat group, but I think we'll let people, because we've got about 5 minutes, let people raise their hand if they have something that they'd like to verbally ask or something that is in the chat that they'd like to elaborate on. So let's see. Alan had his hand up, but I'm not sure if it's left over from last time.
I'll just, it was, but I'll just quickly comment. I didn't realize how to put down that the reproducibility issue is getting to be very, very large, and particularly in ecology and mathematical biology, people are stressing this much more in terms of computation, and so being able to code well and to make your code available via GitHub, etc., is a skill that's really, really important, that maybe is even more important than some of the specific aspects. And so students really need to start to do that.
I completely agree. And we have to prepare the students for the next 25 years, right? So it's not even sufficient what we can get away with. Like, they will live in a different world from that aspect.
Okay. Thanks. Other questions? There were a set of comments and links regarding reproducibility in the chat, so I don't think we need to go back over them. But other comments or questions from people? Jennifer, I see you're raising your hand.
Hi, I just had a comment, which was that I think along with the data science skills, the computing, the math, the reproducibility and that stuff, it's probably also of value to the students to learn how to work in interdisciplinary groups, to talk across disciplines, because I know that students sometimes struggle with that, faculty struggle with that, to be honest. How to understand the common language that people in our discipline take for granted, but when we cross disciplines, I mean, as a statistician, this happens to me all the time, right? You use the word normal, and people are like, yeah, and you're like, no, that means something very specific to me. And we use this graphs, images, sequences. There's lots of language that, I feel like we need a Rosetta Stone of, to help our students to understand so they're effective in those situations. Because, this area already brings together life and data sciences, but to help those students to understand and be able to communicate well. I think that's also part of the training that we need to offer.
Yeah, I forgot to mention, like, these groups that we're having are socially engineered, again, to put them like exactly in that situation where there's no two students in the same group that are from the same program. And we try to ask them, what do you know already? And mix people that know a lot with people that have never opened a computer, right? Exactly to foster. I mean, it's a bit throwing them in the deep end and say, swim, right? So that's why we're doing all these team building activities. And we have like one faculty member that follows each group and meets with them every so often at dinner, saying, how are things going? How do you communicate with each other and whatnot?
Oh, that's great. Stefano, thank you so much. I know that there's going to be more conversations, and particularly about how we might build and take some of your boot camp kind of experience without the funding.
We have the same problem. This year was virtual, right? So you could do it much more cheaply, but I don't think it's the same thing.